fascinating sessions coming up after today, which include biosensors and environmental monitoring, which will be on Tuesday, the 17th of November from 12 to 1:30. And the words epidemiology and pandemic early warning crop up here. So it's sure to be fascinating and topical. We're also going to be having a second session on Tuesday, the 1st of December from 12 to 1:30 on sustainable biological removal of nitrogen and phosphorus, followed by a session early in the new year on organohalide bioremediation. And this session will be on Tuesday, the 12th of January at 9 till 1 till 10:30, because one of our speakers is joining us from the University of New South Wales. And I think Drew can say that the time difference is kind of interesting. Anyway, visit the events section on our website, which is ebnet.ac.uk for registration and more information. And without further ado, I'd like to introduce Dr. Yongchang Liu from the University of Southampton, who has kindly organized today's session and is our chair. Yongchang, over to you, and I'll disappear myself.
Okay, thanks Angie for the introduction. It's our great pleasure to have the Dr. Zhiwu Wang, assistant professor from Virginia Tech, America, as our first speaker to give the presentation. We are incredibly grateful to Zhiwu Wang, because now it is 7 a.m. in America time, so it is hard. Okay, so his presentation title is Aerobic Granules in Continuous Flow Reactor for Wastewater Treatment. So now, Dr. Wang, it's your time.
Okay, let me share my PowerPoint. Hopefully I can do it right. Let me do it in presenter mode. Do you see my PowerPoint?
Yes, I can see.
It's good? All right, thank you very much. So I think good afternoon, UK, and good morning, US, and good night, China, I guess. Today I'm going to talk a little bit about aerobic granulation in continuous flow bioreactor for wastewater treatment. And because I only have like 20 to 30 minutes, so I will try to be brief. And then if we want to dive into more details, we can either do that in the Q&A or we can communicate offline. By the way, even though my name is Zhiwu, but I just go by Drew to make it simpler for everybody. I am leading a Center for Applied Water Research and Innovation in America. This center actually is located here, where it's only like 45 minutes away from the White House. So that means I'm in the greater Washington metropolitan area. And my center mainly serves the application-oriented research for utilities. So I'm covering the research needs of many utilities and also many consulting firms in Virginia, Maryland, and also Washington, DC. And those are the funding agency has been kindly supporting my research here. And I even placed my university research lab in a local utility here called Wastewater for the convenience of the real world research. With that being said, today I'm going to cover 4 questions. Number 1 is why continuous flow matters. Number 2, how to achieve continuous flow aerobic granulation. Number 3, there's lots of controversy over the importance of Fisa and Raman. So I'm going to elaborate a little bit more on that. And finally, I will show 2 case studies how to do full-scale continuous flow aerobic granulation, but that will be very brief for the time limitation. So let's look at the first question, why continuous flow is important in the aerobic granulation world. Actually, all the details actually summarized in this publication. If you have more interest to read it, I'm just going to give you a very brief version here. So if you are interested in granular sludge, most likely you like its excellent settleability and also high reaction rate because of the high biomass retention. And also those granules has layers. It can have aerobic, anaxic, or anaerobic zone in one grain of granule. So basically you have one granule function at 3 bioreactors. So that's very good process consolidation. And biogranules, actually they are one kind of biofilm. So they have the other advantage that biofilm has. For example, they're more tolerant of the shock and toxic loading. And granule also we found they are easier for dewatering. So you will save polymer for your cake dewatering work. And finally, using granule may save lots of money. Actually, this number here, actually from a case study from a utility here in Northern Virginia, that utility called UOSA, and it's Upper Occoquan Service Authority. Actually, they need to upgrade their treatment capacity. Basically, they have two choices. One is they build more tank with more concrete. The second choice is they replace the activated sludge with granular sludge. And they compare the two scenarios. So finally, if they go with the granular sludge, they will save 94% of the cost as compared to building more concrete tanks. But they require this granulation process to be continuous flow rather than sequential batch. Okay. If granular sludge is so great, then I have a big question. I had this question like 2 years ago. The first granular sludge, actually aerobic granulation study was done in like 20 years before. But why after 20 years R&D, there is still rarely any aerobic granulation technique being applied in utilities? So that's a big question I had. To address that question, I wrote this review paper back in 2018. I think that's 2 years ago. I try to summarize what has been done and what is the problem. So here are the main points. First, over these 20 years, mainly, most is like 99% of the aerobic granulation study or application was done in sequential batch reactor somehow. And the complete mix reactor actually somehow it doesn't work for aerobic granulation. So there's no luck in the CSTR reactor. And so far, the major full-scale application was done by Nareda, but still in sequential batch reactor. The sequential batch reactor has its own limitation. For example, most of the wastewater treatment plant or utility use continuous flow system. So if you want to switch to sequential batch, you need to build more, new infrastructure that is more costly. And also sequential batch reactor, we know they are more complicated to operate. And also sequential batch reactor will do the feed or discharge within only like 10% of the HRT of the reactor. Actually, that's a big problem. First, if 90% of the time is not taking in the influent, those influent need to be stored either in an equalization tank or some other parallel SBR reactors. Or, okay, that's problem number one. Problem number two, if you want to feed or drain the water within 10% of the time for 90% worth of the influent, that means your piping and the pumping system need to be like 10 times larger, which is very difficult to implement in reality. So those might be the reason why aerobic granulation technique is still not very popular after 20 years R&D. So I kind of answered the first question, why continuous flow matters. if we can have the continuous flow, it will be a lot simpler and cheaper, more reliable because it has less moving parts and more adaptable to existing wastewater treatment plants. So eventually it's supposed to be more applicable over SBR. So that's what we desire. And again, more details can be found in this publication here. Now, let's move on to question number 2. If continuous flow granulation is so great, how to do it? Why very few people did it? A little bit background about the mechanism of aerobic granulation. Actually, those 2 publications was by me like 15 years ago when I was a graduate student. At that time, all aerobic granulation reactors are SBR. There's no continuous flow. The reason is we believe the SBR is easy to give a settling velocity selection pressure, which actually can be calculated by this formula here. Is the discharge height of the SBR over its settling time. Actually, that gives minimum settling velocity, which means any bioparticle settle slower than this selecting settling velocity, they will be washed out of the bioreactor. That's very important because there is a knowledge point here. Bioflux or tiny bioparticles, they always grow way faster than granules because they are higher specific surface area. Means if we don't get rid of those bioflux, they will quickly outcompete biogranules. So that we believe that was the ultimate driving force for granule to survive and prosper in aerobic granulation reactor. Okay? And this figure just showing if I dial up this settling velocity selection pressure, more granule will be formed, and those granule size will become larger because they actually become more competitive when I remove their competitor. Okay? So this was 15 years ago. Now, the same settling velocity selection pressure can be provided by lamellar plate or hydrocyclone. I will elaborate a little bit more on that. Okay, this is my chain of thought like 2 years ago. So after I did my review, this is the big picture I saw. First, if you combine SBR with selection pressure, if you integrate selection pressure in SBR, you form very good aerobic granular sludge with no problem. That's no brainer. Anybody can do that. But if you integrate selection pressure in CSTR, the single tank complete mix reactor, nobody can do aerobic granulation. So it just doesn't work. No matter what kind of shear force you provide, what kind of FM ratio you provide, it doesn't work. So what is the difference between SBR and the CSTR? Actually, all parameters can be set the same, no matter it's loading, FM ratio, or whatever, except one parameter is always different. That is the profile of the substrate concentration in those reactors. For example, in SBR, there is always a Fisa phase and a Raman phase. In CSTR, it's actually always Fisa phase. So my hypothesis is if that's the major difference between the two, what if I reproduce this Fisa and Raman condition in a continuous flow reactor to see whether it also can form granules? So that's my hypothesis. Of course, that hypothesis has been proven valid by my last year's research. But basically, the idea is plug flow reactor is a continuous flow reactor. Plug flow reactor also provide a Fisa and Raman condition, even though it's in the spatial direction rather than in the time direction in SBR. But basically, they do the same job, Fisa and Raman plus selection pressure. Okay. Here is my experimental design. To mimic a plug flow reactor, by the way, ideal plug flow reactor does not exist in reality. So I have to connect 10 CSTR reactors in series to approximate a plug flow reactor. And I place this flat flow reactor in the local utility, as I said, is Upper Occoquan Service Authority, UOSA. So this utility, actually, they are interested in full-scale aerobic granulation. So I try to set all my parameters same as their full-scale system. By the way, their full-scale treatment chain, secondary chain, has a high FM selector at the very beginning. Actually, that provides a very good Fisa and Feynman condition there. But the major difference between my pellet and their full-scale is here. So my pellet applied a high selection pressure that's like 10 meter per hour, and their full-scale just use a regular clarifier. Actually, there is also a selection pressure on the setting velocity. That's the surface overflow rate, which is only 10 meter per hour. So my selection pressure is like 10 times theirs. That's the major difference between the two. After, by the way, we feed the same water, okay? After like 3 months, very big granule formed in my continuous flow system, but as expected in the full scale, they only have the bioflux there. When I look at the other parameter, I have good news and bad news. The good news is in my pilot reactor, this red line here, they all have very good settleability in terms of the SVI or zone settling velocity. They are super good, okay? By the way, this experiment was done in last winter. It's like December. So the winter time in UOSA actually is a horrible time for the sludge settleability. Usually the settleability become worse when the weather is cold here. So the full scale actually have much worse settleability. So that's the good news for my pellet reactor. Now, bad news. Like almost all aerobic granulation reactor, during the start-up phase, there is like the first month, almost all ml VSS was washed out because the selection pressure. And as a result of that washout, my reactor actually lost almost 100% of the nitrification capacity and also lost like 70% of the COD removal capacity. Actually, this is a bad news because in full-scale application, this can't be accepted. We cannot interrupt our treatment performance during the startup of the granulation reactor. Actually, that's a new research topic in my research center now, and we are doing some work on that, trying to fix that problem. Lots of data, but I want to just draw your attention to 2 data here. If you compare my pellet and the full-scale reactor, the pellet always have higher effluent TSS. It's way higher than the full-scale clarifier effluent. Actually, that's expected because that's how the selection pressure works. It should keep getting rid of those bioflux. And so later we take advantage of this high TSS as a means to control SRT. Okay, I'm going to talk more about that later. And also one thing is the total COD removal of my granular reactor actually is not as good as the activated sludge reactor. The reason might be attributable to the better absorption, larger specific surface area of the activated sludge. So possibly a polishing process is necessary after the aerobic granulation reactor. Okay. All right. So I think I kind of answered the question 2. How to achieve continuous flow aerobic granulation? So we need Fisa-Feynman condition in flat flow reactor in combination with certain velocity selection. So if you want to see more detail, you can refer to this publication here about that part of the study. Now, let me move on to question 3. I have repeatedly mentioned Fisa and Feynman condition, but actually this is a very controversial topic. The reason is most of the Fisa and Feynman study in the field of aerobic granulation study was done in sequential batch reactor. But the problem of sequential batch reactor is you cannot control feast and famine condition as a sole variable. The reason is, for example, in SBR, if you want to achieve, let's say, more feast condition, less famine condition, you either increase the influent substrate concentration or you shorten the HRT. For example, you shorten the cycle time. Okay. Well, look, there is a problem. If you increase the loading rate, actually, that's unfavorable for aerobic granulation, because a thumb of rule in this field is if the bacteria grow faster, they tend to form floc rather than granule. So granule usually form by slow growing bacteria. Okay. On the other hand, when you shorten the cycle time, for example, you shorten cycle time from 4 hours to 2 hours to achieve more feast fraction, you actually also increasing the selection frequency. Means 4 hour cycle time, you select 6 times a day. If you go to 2 hour cycle time, you select 12 times a day. So actually more selection frequency will favor granulation. So somebody changed the loading, that's unfavor. Somebody changed the cycle time, that's favor. So the combination of these two opposite effect will be controversial. So you may not see the real effect of Fisa famine condition. Okay, with that being said, what I'm trying to do in continuous flow reactor is, continuous flow reactor is able to control Fisa famine condition as a sole variable. Okay, this is what I did. I have 3 plug flow reactor here. Okay, they share everything same influent, same retention time. But the major difference is the number of CSTR I connected during that same HRT time. So why I'm doing this? Let's look at this first reactor. I have 8 CSTR in connection. Okay, if I increase this number all the way to, let's say, infinite number, this first reactor will become an ideal plug flow reactor. Okay, we know biogranule will form in plug flow reactor. Okay, the opposite, this third reactor, if I reduce the chamber number all the way to only 1 chamber, that becomes CSTR reactor. We know granular sludge doesn't form in single CSTR reactor. So this first and third plug flow reactor just representing 2 polar scenario. And this second reactor represent the in between, a mild feast and famine condition. So as you can see, I indicated here, when there is much better plug flow condition, the feast phase will be shorter, the famine phase will be longer, and vice versa for the poor plug flow condition here. You already can see the results difference between those 3 reactors. So the 8-chamber reactor actually got very good granulation. The 4-chamber actually only cultivated some big flux. Actually, they have poor settlability. By the way, I forgot to mention all 3 reactors actually share the same settling velocity selection. So everything same except the chamber number used. Now, if I'm studying feast and famine condition, I need to define what is feast and famine condition. Unfortunately, in literature, there is no clear definition of what is feast, what is famine condition. So I have to take liberty to define my feast and famine condition. Okay, basically, what I did is, we know bacteria or even us, while we're growing, we also decay or maintain ourselves.
Sorry, I'm having trouble.
So I define whenever there is growth rate higher than the decay or the maintenance rate, I will call that a feast condition. Vice versa, whenever the growth rate is less or equal to the decay or maintenance condition, I call that famine. Okay. Basically, just like me, I'm trying to lose my weight now. So the way I lose my weight is I try to make sure my the food that I take in actually provide less energy than my maintenance. So that will put me in famine condition. I will lose my weight. So the bacteria, I'm forcing the bacteria to do the same here. So based on that definition, I can calculate the Fisa famine condition in each chamber of my blood flow reactors. For example, if you can follow me, let's look at this 8-chamber reactor. This blue color here, this blue dashed line actually represents the specific decay rate of this reactor. Actually, that's the maintenance rate of that reactor. If you look at the first chamber and the second chamber growth rate, actually they are about this maintenance or decay rate there. So they're supposed to be feast phase. If you look at the third chamber, fourth, fifth, sixth, seventh, eighth chamber, they're all below this decay rate. So I regard them as famine phase. So in that case, 2-chamber feast, 6-chamber famine. So that's the ratio of feast to famine 1/3. So that gives me the feast famine ratio about 0.3. So I did the same calculation for my 4-chamber reactor, 6-chamber reactor. So they all have different feast famine ratio. But as you can see, the smaller this feast and famine ratio is, actually the easier to form granule. So I also plotted all the other granule characteristic information against this feast and famine ratio. The smaller this ratio is, the better settleability, the better circularity or more round morphology of those granule will have, and also the granule will become denser, and the MLVSS will be a lot higher when the feast and famine condition was low. So that just means better granular sludge has been formed and they retain more biomass there. Okay, so I kind of use my experiment to prove the importance of feast and famine condition. And in my world, the feast and famine condition in blood flow reactor and SBR, they are the same. So they're supposed to have the same mechanism. So I analyze all the literature data I can find reporting the feast and famine condition, and also the settling velocity selection they used, and also the final sludge settleability they cultivated. So I plotted all those literature data together with my own data in this graph here, try to find the correlation between these 3 parameters: the feast and famine ratio, the settling velocity selection, and the SVI. So this is what I found here. With those data, we can find whenever the feast and famine condition, let's say, is more than 0.5, the SVI will always about 60, which is not good. So no matter how I dial up or dial down this settling velocity selection, it doesn't help the SVI. So this right zone is more like the CSTR. So no matter what you do, granule not supposed to form there. And there is a green zone here. It's between like 0.25 and 0.5. Actually, my 8-chamber reactor was in this zone. The feast famine condition in my 8-chamber reactor was like 0.3. So it's like here. Means if I want to go to this green zone, I need to dial up the settling velocity selection pressure. Actually, what I did, I picked a 10 meter per hour. Actually, it's way up there. So it's supposed to be here. Means I can achieve SVI less than 60. But here is the interesting part. The data also indicating if we are able to dial this feast and famine condition all the way down to, let's say, 0.1. In this blue zone, the settleability will become insensitive to selection pressure. Actually, this kind of in line with lots of reports from utilities saying they don't have very high selection pressure, but whenever they have a very good plug flow system, they tend to cultivate much better settleability sludge. So that's kinds of falling in this blue zone here. Anyway, this first figure just showing us this Fisa ratio is very important. And when this ratio is low, we do not need very high selection pressure. When this ratio is in this middle zone, we need the help from the selection pressure. When it's in the red zone, even the selection pressure doesn't help. Okay. And this second figure just telling us how to implement this mechanism into a real wastewater treatment plant. So if you have a wastewater treatment plant, you want to grow aerobic granulation sludge there. And then the first thing is you need very good Fisa environment condition there. There are mainly two ways to change that. One is you increase the chamber number by adding in the baffles there, or you can change in some traditional way. You create a better FM ratio zone at the very beginning, or you change the HRT or whatever. But okay, this is simpler strategy. Okay, if you can change the chamber number, actually that's simpler than change FM ratio or the other parameter. So what I propose here is there is a Dunkler number. Actually, that's a classic chemical engineering number. So that number actually it includes FM ratio, retention time, all those parameters in this one parameter. So this Dunkler number actually, as you can see, is the product between hydraulic retention time and reaction rate coefficient. Basically means if this Dunkler number is high, your Fisa time will be relatively longer. And if this number is high, your Fisa time will be relatively shorter, and ferment time will be relatively longer, okay? Because the reaction was faster there. So any wastewater treatment plant, you can calculate your own Dunkler number here. For example, if your number is 30, and if you want to achieve a Fisa ferment ratio, let's say 0.25, which is an ideal ratio, you will see from this graph, you can see 10 chamber number is required. That means you may want to modify your secondary treatment chain by adding in more baffles to create 10 chamber numbers. Okay. All right, so I kind of answer the question 3. How important is the Fisa ferment condition? My answer is there is a threshold. You don't want your secondary tank have this Fisa ferment ratio above that threshold. If it's above that number, the granulation will be very difficult to achieve. And the lower that Fisa ferment ratio is, actually the less help you need from the settling velocity selection. So the Fisa ferment and settling velocity selection pressure, they complement each other. Okay? So if one is higher, the other can be lower. All right, how much time I have? Okay, I already run over time, sorry. Let me quickly wrap this up. Next is how to wrap this, how to do continuous flow aerobic granulation in full scale application. I brought 2 case studies here. One is to modify your side stream. The other is to modify your main stream. So I'm going to walk you through each of these 2 case studies. So the first one actually was done by Jacob in USA, the same wastewater treatment plant where I did my pilot scale aerobic granulation research. So this plant, as you can see, it's symmetrical, has 2 chains. So what they did is, what the Jacob design was, they installed the hydrocyclone on the side stream. So basically they selecting a portion of the rats to get rid of the bioflux from there. That's also where they control the SRT by the overflow from the top of this hydrocyclone. So from there, they control the SRT around 16 days. And the reason why apply hydrocyclone on the side stream rather than in the main stream is because hydrocyclone doesn't have the capacity to process high flow rate of the water. So side stream has less flow rate, so it's easier for hydrocyclone. So when they compare the train with hydrocyclone and to the train without hydrocyclone, actually the hydrocyclone side showed very much better sludge settleability even during the winter time. That's last December. Actually, that's the worst time for the settleability in this plant. Oh, by the way, I forgot to mention this plant, they have a high FM selector at the very beginning. So actually, we have already confirmed this secondary chain has very good Fisa famine condition there. So what they need is just a selection pressure. So they did that in the side stream there. The second case study was done by AECOM in a wastewater treatment plant called Penticton, I believe it's in Canada. And they did this by modifying the main stream through the installation of a lamella plate. Lamella plate can process high flow rate of the water, so they can use that with no problem in the main stream. Basically, they put that in front of the secondary clarifier. So they also compare, oh, by the way, this treatment chain, actually they're doing the BIOP and the BNR at the very front of the secondary train. So that gives very good Fis condition there, and the aerobic zone, there are many famine condition. And when they do the BIOP, actually they did the primary sludge fermentation to provide a carbon source for BIOP. So that's even better Fis condition there. So what this plant need is just a selection pressure that's provided by the lamella plate, and that's in the main stream. So the results, as you can see here, the side with the lamella plate give very good cyclability, but the other side without it, selection pressure actually got very poor cyclability there. So that's how they did this in a full scale application. So my answer to the question 4 is, in order to achieve continuous flow aerobic granulation in full scale application, we need to make sure, or we need to provide very good Fisa famine condition in the main stream. As I have shown you, you can change the chamber number there by adding baffles to achieve this. And also, you need a certain velocity selection pressure, which can be provided either by a lamella plate or hydrocyclone. And even somebody proposed the high ISOR clarifier that should also work, but we haven't verified that yet. All right, with that being said, this is the end of my talk. I really appreciate the data and the financial support from UOSA, Jacob, and AECOM. Without their support, there is no way I can do those studies. And thank you for your time and interest. If you have more questions that I cannot answer during the Q&A, we can communicate offline. Thank you very much.
Thank you very much. Thank you, Dr. Wang, for this interesting and brilliant work. I think that we have a couple of questions coming to the chat. Can you explain a little bit how granules are formed? Because some audience are not familiar with the granule technology.
Okay, how granule were formed? That's a very good question. That depends on what level of answer you're expecting from me. Okay, molecular level is still a mystery. Actually, what we usually find is the granular sludge actually doesn't have major microbial community change as compared to the activated sludge. So what we only have hypothesis at molecular level, that is the same bacteria, they show different phenotype. So under selection pressure, so the very convenient explanation is under selection pressure, the bacteria will fight to survive. So how they can survive in this reactor to avoid being washed out is they excrete the bacteria glue. We call it extracellular polysaccharides. Actually, people also call it EPS, extracellular polysaccharides, or here is polysaccharides or protein. As you can see, the pilot reactor with lots of granule formation there, they have much higher polysaccharides and protein excreted to their extracellular space as compared to the activated sludge. So the bacteria will respond to the selection pressure of Fisa condition we provided to produce the glue, to glue themselves up, and then to help them survive in this reactor. Okay. Anyway, that's not very in-depth explanation. As I said, the molecular level is still a mystery now. Maybe Jeremy can provide more insight in there.
Okay, thank you so much for your explanation. We still have a couple of other questions, but because of the time constraint, we move to the second speaker first. And after the second presentation, we probably can collect the question and answer together.
Okay, let me stop my sharing here. All right, I will mute myself.
Okay, it's our great pleasure to have the Jeremy Webb, a full professor of Professor of Microbiology at the University of Southampton as our second speaker. He is also the co-founder of the National Biofilms Innovation Center. Probably he can give a little bit more detailed introduction about that center. His presentation title is Molecular Toolbox for the Understanding and Control of Complex Microbial Consortia. Let me hand over to Jeremy, please.
Thank you. Could I check that you can hear me and that you can see my presentation?
Yes, yeah, I can see your presentation.
Excellent. Okay. All right, thank you very much for joining this online session. And I'd like to thank the organisers and Young Chang from EBNet for inviting me to give a talk. I want to talk about our research and interests and some of the activities within NBIC focused on understanding and controlling biofilm development. And in doing so, I hope to draw some relevance to environmental biotechnology processes with the intention to cross-fertilize ideas and discussion across both biofilm research and environmental biotechnology research. I take it everybody can see the presentation. Yes, okay. So when we study biofilms and we look at their development, we see structured communities that form aggregates of cells, often quite an exquisite architecture with cells embedded in a self-produced sort of polymeric matrix we were just talking about just now. We see this in the laboratory, but we also see it in other environments. In a clinical context, biofilms cause very problematic, cause very difficult to treat infections. But in an environmental context, then we see, for example, shown here, sort of epiphytic biofilm aggregates on the surface of a marine plant, that example, but also, of course, aggregates that are exploited in an environmental biotechnology context, including granules. So microbial aggregates, are part of bioprocesses, and it's the aggregated nature of the microbial growth that is heavily depended upon for practical applications. So I want to skim over some of the tools that we have for biofilms that allow for sort of detailed exploration and understanding of biofilms, and I hope that perhaps some of the techniques that I'll cover and that perhaps are being developed in other application areas, possibly biomedical or other, may have may stimulate some discussion and or have application potentially in environmental biotechnology too. So the way I want to structure this talk is to firstly, consider some of the existing and emerging approaches to study community composition and architecture and function. And then secondly, to look at a few of the processes that may influence the regulation and development of community processes and structure of relevance to biofilm and aggregates. In other words, to consider some of the underlying biological processes and some of the molecular mechanisms that we were just alluding to earlier as well, that may have an impact on aggregate formation and dispersal. Lastly, then, to considering approaches to understanding community composition. So in terms of who is there and what they're doing, of course, I need to give brief mention, as brief as I can, to the approaches for understanding microbiomes. And of course, the scale and the power of omics approaches, continues to increase all the time. So commonly, 16S PCR and sequencing of the ribosomal RNA gene and its divergence. That's quite a common, and it's relatively cheap and convenient way to profile a microbial community. That will give you a taxonomic profile, but of course it won't capture the sort of broader genetic content or capability and function within that community. So for that, of course, whole metagenome sequencing the entire, fragmenting and sequencing the entire genetic content of a microbial community is much more powerful in understanding sort of genetic and metabolic capability. Again, huge advances in this area. For example, it's now possible to take an environmental DNA sample, and from that to build and assemble finished, complete bacterial genomes from metagenomes. So to assemble entire completed bacterial genomes from an environmental sample. And these kinds of technologies are substantially advancing microbiome and microbial ecology research. There's many challenges in this area, of course. So establishing correlation versus causation can be very difficult. It's one thing to see a shift in a microbial community or patterns in genes, but to establish mechanistic understanding of why that shift has occurred can be very difficult. And likewise, standardization and quantitation of omics data can be very problematic. You have different protocols and sequencing platforms that each might introduce their own biases. So it can be very difficult to control for these and standardize for different technologies. But nevertheless, omics technologies are leading to exciting new ways to work across big data, metadata, and artificial intelligence, and it's becoming possible to integrate and harmonize large complex data sets, improving the understanding of which sort of mechanisms are driving the biology of the system. This is leading to approaches such as pathway analysis and profile biomarkers. In other words, it's possible to, for a particular phenotype or experiment or bioreactor that you're looking at, to see the entire network of regulatory pathways and to visualize that data in clever ways like topological data analysis. So you can visualize these sort of network patterns that can characterize a particular phenotype or experiment. So I think this sort of data integration is becoming much more advanced, from the transcriptome to the proteome to the metabolome, all sort of mapped and integrated together. So I think there's a huge potential there for whole systems understanding of environmental biotechnology processes. Just to give an example of some of the work we're doing in this area, Joe Parker is one of the National Biofilm Innovation Centre research fellows. So Joe has been working in the area of sort of real-time sample characterization and sequence analysis. So he's working at the sort of point of application, if you like. So he's been pioneering sort of field-based preparation at the point of application of your DNA samples, and then providing real-time sequence information using protocols and software and cloud-based tools based around this sort of USB plug-in MinION sequencing platform. And I suppose the objective of what Joe is trying to do really is to, well, firstly, to obtain DNA at the point of application, but also I think the key here is that, you know, a high level of bioinformatics sort of expertise is not always accessible to operators or engineers, or and how do you sort of make sense of all that omics information? And the idea that Joe is keen on developing is to make to provide sort of real-time information that can be useful, you know, and helpful, you know, information about the key organisms or functional groups that might have an important role in the system, and to provide that information in the simplest way possible to decision makers without the burden of all the sort of complex bioinformatic sort of pipeline to wade through. So he's doing this at the moment for clinical applications, but there's no reason why this kind of approach can't be used to sort of give real-time understanding of bioprocessing, you know, applications. So of course, everything I've talked about so far relates to DNA or RNA extracted from whole sort of homogenized microbial communities. So often, you know, the function is sort of implicit in the structure, right, of the microbial community, and so this sort of DNA sequencing approach doesn't really give structural information or information about the spatial organization of microorganisms. So for this, we need imaging approaches. So fluorescence in situ hybridization, or FISH, is not a new technology. It's been around for many years, and that's used to study the structure of aggregates. It's been used in environmental biotechnology. Here it's distinguishing bacteria and archaea in a thermophilic granule example, and also distinguishing polyphosphate accumulating organisms from the rest of the bacteria in another example here. The problem with FISH in this context is that, you know, typically the filters and the bandpass filters for conventional fluorescence or confocal microscopy will limit the number of colors or fluorophores that you can use. So you're often sort of stuck with 2 or 3 microorganisms that you can target, or groups of microorganisms, and it becomes very difficult to get more complex than that. There are some more advanced FISH approaches. CLASI-FISH is, you know, combinatorial labeling and spectral imaging, gets around that problem that I'm using combinations of probes together with sort of spectral imaging that can scan across the entire spectrum. So you can, with a very small number of fluorophores, that can be used to tag 10 or 20 or more organisms within a community, and this has been used to good effect here in this study of sort of biogeography of microorganisms in the human oral microbiome. So it'd be interesting to apply these kinds of approaches to some of the granular structures that we've been looking at. But of course, you know, targeted microscopic labeling approaches like these, they're by very nature, they're extremely specialized and labor intensive, and they require a lot of processing and optimization of techniques. So we're interested in some label-free imaging technologies that can provide information. It can provide information on sort of both the spatial structure as well as the composition, potentially without sort of, you know, all that intensive sample processing. So Raman spectroscopy and imaging. So Raman spectroscopy is a direct label-free spectroscopic technique, which is used to characterize a sample by its molecular and sort of vibrational fingerprint, if you like. So in microbiology, it's been used to differentiate bacteria at the species and strain level. It can do that. You know, we've been using Raman spectroscopy in the diagnosis of, you know, in infection scenarios, clinical scenarios, where, you know, rapid and specific detection and identification without the use of labels, you know, can lead to, you know, faster and improved clinical outcomes potentially. So it's possible to, so these are respiratory pathogen examples that we're looking at here, but just it makes the point that it's possible to separate and identify organisms based on their Raman spectra. But you can also do imaging with Raman as well. So coherent anti-Stokes Raman spectroscopy is a technique capable of using the data obtained by Raman spectroscopy and imaging that data in 3 dimensions. And this has been explored in the fields of cancer and neuroscience. So there's an example here on the left, which is from a tumor, I think, and you can, you know, the chemistry of that tumor can be visualized. The chemical components can be visualized and given a pseudo color. So there's no labeling as such, but you can distinguish collagen, lipids, water, proteins, and potentially other molecules, but, you know, based on the Raman vibrational signatures. So it's been explored in a number of fields, but not hugely in microbiology yet. And so what we're looking at on the right here is perhaps one of the first CARS images of a biofilm. In this case, we're looking at the lipid content. Again, this is direct imaging, no labels, but this is showing the lipid component of the biofilm. The red is actually autofluorescence in the background, but it's giving us information about the chemistry of the biofilm. And these techniques can enable rapid and label-free and identification of bacteria within biofilms with no processing, including directly from samples. So I think there is potential here for, you know, site and real-time monitoring of granules with these kinds of technologies. Okay, so coming to the second part of my talk, looking at some of the developmental aspects of biofilm formation and dynamics that might be relevant to environmental biotechnology. So if aggregation and disaggregation of the component microorganisms is a key feature of environmental processes, then of course we need to understand some of the underlying biological sort of developmental processes that regulate this. I think that's important. And biofilm development is heavily regulated. There's a life cycle sort of component to it. We've studied the development of biofilms for many years, and in my group we've had a particular focus on mechanisms that might lead to disaggregation or dispersal of established biofilm structures. I'm just going to talk a little bit about that, sort of some of the active dispersal mechanisms that can occur in biofilms. You know, being confined for bacteria, being confined within a microbial aggregate it is not the endpoint of development, if you like. You know, there are active mechanisms by which bacteria can disperse from biofilms, and we very often see, you know, in biofilms that these sort of hollow, emptied out aggregate structures where the cells are just gone from the center of those structures, and you can see some of these dispersal mechanisms happening here. So we found that well, it's well known actually now across you know, within biofilm research broadly that cyclic di-GMP is a really important regulator of biofilm formation. This is a sort of intracellular sort of second messenger molecule, but if you have high levels of cyclic di-GMP inside cells, it tends to promote biofilm formation and aggregation. And if you have low levels of cyclic di-GMP, then you would tend to get biofilm dispersal and reversion to the sort of planktonic lifestyle, if you like. And we found some years ago that nitric oxide is can modulate the levels of cyclic di-GMP in bacterial cells, and we spent a lot of time unraveling the mechanisms by which that occurs. But nitric oxide can modulate cyclic di-GMP, and when we add back nitric oxide to biofilms in the form of nitric oxide donor chemistries, then it's possible to disperse biofilms using very low concentrations of nitric oxide donor molecules. These are non-toxic concentrations of nitric oxide donors, many can trigger dispersal responses in bacteria. We work a lot with Pseudomonas aeruginosa, but there are many bacteria that have cyclic di-GMP sort of signaling pathways. So we try to understand this process, and we are applying some of this technology from a clinical perspective again, but also, you know, from an engineering perspective. I just wanted to mention, you know, a pilot trial that was carried out by our partners in Singapore, the Singapore Centre for Environmental Life Sciences Engineering, and they were interested in removing biofilms in this context from a reverse osmosis, you know, a fouled reverse osmosis system. And they found that by adding low concentrations of, a nitric oxide donor into the sort of clean in place fluid solution, that they were able to, you know, substantially improve the cleaning process and induce detachment of biofilms in that reverse osmosis membrane system. So I think it would be interesting to understand more about CDGMP and you know, and how its levels might impact on granulation processes. But there are also, likewise, there are other signaling mechanisms, including quorum sensing, that I think represent areas of opportunity across different sectors potentially. So quorum sensing, through its impact on biofilm regulation, you know, I think has a potential application in a range of settings, clinically through to biotechnology applications. Quorum sensing enables bacteria to control expression of specific genes in relation to cell density, or the cell densities at which the resulting phenotypes will be most beneficial to the bacteria. So one of our NBIC founding partners at Nottingham University, Miguel Camara, works extensively in the molecular and regulatory pathways involved in quorum sensing. They're targeting infection as their sort of principal application, and they've been developing inhibitors of quorum sensing, in this case the quinolone quorum sensing system of Pseudomonas aeruginosa, and they use drug discovery platforms to do that. You know, sort of quite advanced. They develop compound libraries. They use in silico sort of docking predictions and bioreporter systems and structure activity relationships to you know, validate whether some of their inhibitors can, you know, interact with the quorum sensing target. And they use, you know, crystallography to do that as well. I guess the key thing is that they've seen that these inhibitors of quorum sensing, they can interfere with the process of biofilm development. You know, they can, in a clinical context, they can prevent virulence and they can sensitize biofilms to antibiotics. But importantly, I think these, you know, when you interfere with quorum sensing processes, you also change the architecture and the structure of microbial communities, polymicrobial communities. And the example here is 3 clinically relevant organisms, but you can see that the biofilms are very different in the presence of a quorum sensing inhibitor to you know, to the wild type version. So there's obviously a role for quorum sensing in the architecture of polymicrobial biofilms. And this is seen across a range of signal mechanisms. Just to mention very briefly another signaling pathway, the team at Southampton worked with another signal, the diffusible signal factor, DSF, which is unsaturated fatty acid signal molecule. And again, similarly, the team have synthesized a library of analogues, and several of these analogues will interfere with the signal molecule and can impact on biofilm development and biofilm sort of physiology. Importantly, also, these molecules can impact on biofilm structure, architecture, and development. So I think there is looking, you know, I've had a sort of brief look across the environmental biotechnology literature, and it does seem that there is sort of tantalizing evidence that some of these signaling mechanisms like cyclic di-GMP and quorum sensing do have relevance in the biotechnology processes. It's clear that this signaling is relevant for complex microbial communities, and it's therefore likely that these processes have important functional roles in granulation and activity that we would need to understand. So finally, I'm trying to skim through things as quickly as I can. I just wanted to talk briefly about bacteriophage, because we've worked with bacteriophage in biofilms a lot, and we find that they're very highly upregulated in biofilms. Excuse me. We find that bacteriophage can contribute in many ways to the biofilm development. They can induce lysis of subpopulations of cells within biofilm, but they do seem to have a very interesting sort of role in biofilm survival and adaptation as well. You know, for example, we characterize one particular bacteriophage from Pseudomonas aeruginosa, and we found that it actually forms almost part of the backbone, the structural backbone of the biofilm. It becomes part of the matrix, and it's a filamentous phage that really helps to sort of strengthen and enhance survival of the biofilm. And there's been quite a bit of research follow-up research on this phage, that it can promote sort of crystalline structural properties of the biofilm matrix, which help it to protect the bacterial cells, and it can also contribute to protecting against antibiotics, and can promote virulence in bacteria in clinical context as well. So we're finding that bacteriophage can have all sorts of interesting effects in complex microbial communities. And I just wonder how much is known about because these bacterial viruses play such an important role in the sort of turnover and dynamics of microbial communities. I wonder about their role in environmental biotechnology as well. And I look at some of these pictures of granules, and I see very often, you have a dense layer of cells around the outside of these structures, but often very few cells in the middle. And I wonder, sort of, bacteriophage lysis may play a role in some of that sort of physiology. And I've seen some evidence in the literature that bacteriophage can contribute to the structural properties of granular sludge as well. For example, by releasing extracellular DNA, which becomes part of the sort of polymeric matrix, if you like. So I think that's another area that is sort of under-researched and would be helpful to understand more about in a biotechnology context. So much of what I've been talking about relates to biofilm engineering. How do we harness that grand challenge of how do we harness microbial communities for benefit? This is a question for MBIC as well. We've held an academic industry workshop targeted at biofilm engineering, and exploring the unmet industrial needs, and trying to define what really the sort of key challenges are in some of these areas. And so this report from that workshop is available to download. Please have a look at that. Just to conclude, I think there are many potential opportunities, at the interface of what we do in biofilms and environmental biotechnology. So I hope this presentation at least provides some food for thought and potentially some collaborative opportunities as well. That would be great. And just very quickly, I'd like to acknowledge so the people that do the research. I've mentioned the MBIC fellows, Joe Parker, Callum Highmore, and Shertzi Ahn, and of course, this fantastic MBIC team that we have, the co-directors Kate McPhee, Rasmita Raval, and Miguel Camara, and our CEO Mark Richardson, and operations director Joe Slater Jeffries as well. So with that, I will finish. Thank you very much.
Professor Webb, thank you so much for your excellent presentation, very insightful. We have one question here for you, for biofilm. Can you explain a little bit if the biofilm formation from the different scenarios is different? For example, from the clinical scenario for wastewater treatment plants, is the mechanism of the biofilm formation is the same or different?
So I think that's really the question at hand, isn't it? We understand many of the mechanisms by which biofilms form, in the laboratory and clinically, and we've got quite a, there's been quite a lot of sort of dissection of that knowledge, quite an in-depth understanding of biofilm and molecular processes involved in biofilm development now. So really, the question is whether those are relevant in complex consortia like granule systems, and I think that's what we need to learn. And I think the preliminary evidence suggests that these processes are relevant in complex microbial consortia. So cyclic diGMP does seem to be sort of quite well conserved across a broad range of organisms as a regulator of biofilm formation. So I think it's quite likely that molecule is important in whether or not bacteria aggregate in a particular context or disaggregate. and quorum sensing also. And so I think you know, we're all of these sort of mechanisms that have been dissected in sort of single organism models now need to be understood in more complex polymicrobial systems to understand the sort of the broader relevance of them. But I think fundamentally that the processes of aggregation and dispersal are, you know, you can draw a comparison and parallels between the model systems that we use and granular reactors, I'm sure.
Okay, thank you for your answer to this question. I think another question to Dr. Wang is, what do you think about mathematical models of biofilm or granules flux as individual-based models?
That's actually a very broad question. I think the function of the model is, first, it can predict something. Two, the model can verify a hypothesis or something. So it depends on where you want to apply a model. But one point I want to make is, whatever model need to be verified first. That means you need to have a fundamental understanding before the model development. And then the model verification and prediction will help further verify your experimental results. So model is in between, it's not the beginning of the research process. Yeah. I don't know whether that answered that question or not.
Okay, good, thank you. I think another question to Professor Wang, if a quorum sensing molecule now producing a bacteriophage therapies ends up as a pharmaceutical waste, then would you predict the effect if wastewater treatment systems?
If they end up as a pharmaceutical
waste.
Oh, I see what you mean. If they end up accidentally,
they probably end up in the wastewater treatment plants.
So you could they as a contaminant?
Yeah.
You end up. I think the concentrations that we are looking at when we're using nitric oxide, for example. These are extremely low sort of physiological signaling relevant concentrations. And so I think, you know, well, nitric oxide, you know, is particularly reactive. And to be honest, I don't think that would survive the sort of environmental exposure, and you'd have to apply it directly, I think, to the system for it to have an effect. Quorum sensing molecules, you know, I don't know how long they would persist in the environment, but again, I think by the time they got to a sort of wastewater treatment system, I think they'd be, my guess is that they'd be too dilute at that point to have an impact on microbial community function. I might be wrong, but that would be my sort of intuition, at least.
So yeah, I guess that for the pharmaceutical waste, their treatment is different from the general municipal wastewater treatment plant. So yeah, so I guess even there's damage, probably that the concentration is very low. Okay.
Yeah. Okay. It's all about the concentration, basically. Yeah.
Dr. Wang, there's another question to you. Is there any full-scale continuous flow aerobic granule technology?
Okay, that's a very good question. Let me tell you something that people usually don't pay attention to. Industry, they doesn't care whether it's granule or sludge. So as long as it has a very good settleability, they're happy about it. So with that being said, if you ask whether there is very good settleability, continuous flow, full-scale application, yes. For example, I've shown the AECOM and the Jacob full-scale study there. But the sludge they cultivated may not look like the traditional clear boundary, smooth, round, granular sludge. So if you want those clear looking, smooth, dense, granular sludge, I haven't seen that in full scale continuous flow system yet. So that's my answer. So if you don't care about the looking, you only care about the settlability, there are full scale application there.
Okay, so you have done the excellent work that you have successfully demonstrated that the plug flow reactor can produce aerobic granules as long as the Fisa or selection pressure is used. So what is the next challenge, do you think, that for the further development of this technology for real application?
Okay. My next focus is to find simpler selection and return method. So the sludge return. For example, one reason the full-scale application, they don't produce the ideal looking granular sludge is they're just using the conventional pumping the rice back, and that will for sure break the structure of the granular sludge. So in my system, in my small pilot system, I can easily return sludge with the air lifting method. But how to achieve that in full-scale application, that needs some research there. And also the selection pressure. So Fisa, I don't think there is any major challenge there. But the selection pressure, if we rely on hydrocyclone or rely on lamellar plate, yes, that can have the job done. But is it possible to retrofit the existing clarifier to do the same job? That's even better. So anyway, my research is always to make more complicated things simpler rather than the other way around. So that's my next step.
Okay, thank you. So the question is here is why not get rid of the first stage? Let the granulation rely on the Fisa only. Probably
Okay.
There's a little bit of misunderstanding here.
No problem. I knew this field can be confusing. So Fisa phase allow bacteria to eat the dirt, and Fisa phase allow them to let's say either excrete EPS or whatever. So Fisa and Fisa have different function. So if you want to remove, let's say, COD, you need that Fisa phase. Okay, but on the other hand, you don't want to keep those bacteria too happy. If they're too happy in the Fisa phase, they will not co-aggregate. So we need that Fisa phase. So we need both.
Okay, thank you. Professor Webb, there's a question to you. If I want to induce biofilm formation under anaerobic condition, what advice would you give me?
Is that a question for me or for?
Yeah, question to Jeremy.
Oh, just Jeremy, okay.
Okay, that's a very good question. I think we'd have to set up some experiments in an anaerobic chamber and look at some of the processes that I've been talking about for their impact on promoting aggregation and biofilm formation under anaerobic conditions. I'm afraid I haven't worked in anaerobic microbiology a lot, but it's something we could do, and it's something that we could experiment with, and I'd be interested to look more into that area.
Okay. Zhiwu, another question to you. You haven't mentioned the UASB reactor. Why?
Okay. UASB, right? So yeah, that's a classic granulation reactor for anaerobic granular sludge. But it may not be ideal for aerobic. The reason is aerobic, you need aeration. So there is lots of bubble lifting up the sludge. But in anaerobic UASB, actually, you don't have that aeration problem. But as I remember, Yongqiang have done lots of airlift reactor that function similar way at UASB. So anyway, UASB is mainly for anaerobic granular sludge. That's my answer. Quick answer.
Yes, thank you so much. So I think that I have one last question to Jeremy. You give a very excellent presentation, especially you give the insight to engineers. You know that I'm working as an engineer. So when we develop the new process, new technology, we mainly focus on reactor design, operation optimization. And it seems the microbial study is a little bit missed. For example, so far granular sludge has been developed for more than 40 years, but the granulation mechanism is still unclear to us. If the mechanism is unclear, it is impossible for us to control the granule size, density, microbial activity, and even the long-term structural stability. So what's your opinion about the application of the techniques, the tools developed in traditional microbiology or in the clinical biofilm to the environmental biotechnology? And how expensive it is compared with, because we know that we can use very expensive tools to do the clinical research, but for the environmental engineers, we are treating the waste, so probably it is a little bit different. What's your opinion about this?
Well, I think everything that you've just told me, Yongqiang, suggests that we all should be collaborating. We should be doing interdisciplinary research. It seems that there is a real gap in understanding of microbial mechanism and function in detail in environmental biotechnology processes, and to have that sort of holistic, sort of whole systems understanding of that process, working together, engineers and microbiologists, I think could be very, very powerful and lead to some really new insights. I mean, with regard to whether the technologies or some of the treatments that we're using are can be applied in engineering contexts. Well, I think, as with anything, you have a sort of development phase, don't you? But for example, some of the Raman technologies, it's quite expensive, to do the research initially, but then you would then go into a process of miniaturization or making it accessible on site or useful in an engineering context, and so you start in the lab and then you make it. You apply it. You find ways to apply it in a more practical context, and that. So I think we just need to collaborate.
Yes, yeah, of course. So I think it is 1:30 already. Here we have the last question. Probably that is to Dr. Wang Zhiwu. How can you induce cyanobacteria biofilm considering they don't produce as much EPS?
I'm sorry, I don't have an answer for that because I never worked with the cyanobacteria before. I'm just sorry, I don't know very much about that. But if I can say a little bit more about the UASB, if I can circle back to that question, UASB actually is an upflow plug flow reactor. And the upflow velocity in the UASB actually is the selection pressure, just like in the aerobic granulation reactor. So the point I want to make is the mechanism between anaerobic and aerobic granulation is supposed to be the same, even though we don't have that fundamental molecular mechanism answer yet. Okay, anyway, that's all I can say.
Okay, thank you very much. So that is a very good discussion. I think that is a time for us to end this webinar. I had this to Angie. Angie, do you want to say something?
Yes, I'd like to. Well, I'd just like to thank you all so much for coming today, and the recordings will be available upon our speaker agreement. Louise and I would like to thank you, Yongqiang, for chairing the sessions and for your organization, and special thanks to these our speakers for the fascinating presentations and very much highlighting that broad area where we can sort of help with specific issues like these aerobic granules. We'll be sending an online feedback form, and we'd be really grateful if you can fill it in. It does help us to frame our activities for the network, and it's your network. So, you know, any area of interest, please do contact us. We hope you're able to join us for a future event, and we hope to see you soon, either virtually or in real life. Thank you ever so much, everyone.
Thank you.
Cheers.